Robert's daughter brought a hand dynamometer to Sunday lunch. She pulled it out of her bag between the main course and the dessert, put it on the table, and said, "I want you to squeeze this as hard as you can." Robert, 67 years old, the retired engineer from our second video, the one who had started eating kefir and fermented vegetables 3 years ago after his daughter explained what was happening in his gut between the ages of 56 and 60. Robert looked at the device, then at his daughter, then back at the device. "Is this a test?" he asked. "Everything I bring to Sunday lunch is a test," she said. He squeezed it, both hands 3 times each, 30 seconds apart. She recorded the numbers. And then she told him what they meant, not just for his grip, but for the organ sitting 3 feet above his hands. I'm Dr. Ben, and in this video I want to tell you about the single most accessible and underused cognitive risk predictor in aging medicine. Something that takes 30 seconds. Requires no blood draw, no appointment, no fasting, no imaging, no neuropsychological testing. Just a squeeze. The research behind it is more substantial than most physicians realize, and the mechanism that connects the strength of your hand to the health of your brain is more direct than you would expect. Let me start with the numbers, because they are the part of this story that stopped Robert's daughter mid-sentence when she first read them. In 2022, a study using data from the UK Biobank, the massive British prospective cohort involving nearly 500,000 participants, examined the relationship between hand grip strength and dementia risk over a follow-up period averaging more than 9 years. The sample size, 466,788 people. The finding, individuals in the lowest quintile of grip strength had a 72% higher risk of incident dementia compared to individuals in the highest quintile. And they had an 87% higher risk of dementia mortality. 72%. 87%. Published in the Journal of Cachexia, Sarcopenia and Muscle. Independently confirmed in a separate JAMA Network Open cohort study of UK Biobank participants, which found that lower hand grip strength was associated with increased dementia risk, lower cognitive test scores on fluid intelligence and prospective memory, smaller total brain volume, more white matter hyperintensities, and smaller hippocampal volume. Not just dementia risk, brain structure itself. Now, before I give you the mechanism, I want to be precise about what these studies do and do not show. These are observational studies. They cannot prove that low grip strength causes dementia. There are multiple possible explanations for the association. People in early, pre-symptomatic cognitive decline may already have reduced grip strength as a consequence of the same neurological changes producing both outcomes. Grip strength may be a marker of overall physiological resilience. The same factors that protect the brain also protect muscle function. But here's why that doesn't dismiss the finding. Whether grip strength is a cause, a consequence, or a shared marker of the same underlying biology, it is consistently and robustly predictive. A meta-analysis of 16 observational cohort studies confirmed the inverse association between grip strength and cognitive decline across diverse populations. The English longitudinal study of aging found that grip strength loss rate, not just grip strength level, but how fast you're losing it, predicted the subsequent rate of cognitive decline. Studies have associated lower grip strength specifically with hippocampal atrophy and white matter lesions on MRI. This is not a weak, unreplicated signal. This is one of the most consistently replicated findings in cognitive aging research. And it can be measured on your kitchen table. Now the mechanism. Because there are several, and they connect back to almost everything this series has covered. The first and most direct is what researchers call the neuromuscular connection. Grip strength is not just a measure of hand muscle mass. It reflects the integrity of the entire neuromuscular pathway, from the motor cortex, the frontal lobe region that plans and coordinates movement through the spinal cord, through motor neurons, to the muscle fibers themselves. When that pathway degrades, both grip strength and cognitive function decline. They are two expressions of the same underlying system integrity. The second mechanism is shared biology. Many of the same processes that accelerate cognitive decline, chronic systemic inflammation, oxidative stress, vascular dysfunction, poor cardiometabolic health also accelerate sarcopenia, the age-related loss of muscle mass and strength. Robert had spent 3 years addressing the gut dysbiosis and circadian misalignment his daughter had first explained to him at a different Sunday lunch, and those same upstream interventions had been, quietly, also protecting his muscles. The gut dysbiosis that produces systemic inflammation damages both muscle and brain. The circadian misalignment that impairs glucose metabolism also impairs muscle protein synthesis overnight. Oxybutynin's anticholinergic burden reduces muscle coordination and brain function through the same muscarinic blockade. The systems connect because they are ultimately the same biology. The third mechanism is what some researchers call the myokine hypothesis. Muscles are not passive tissue. When they contract, they secrete signaling molecules called myokines, and some of these myokines, including a compound called irisin, cross the blood-brain barrier. Irisin has been found in animal studies to increase BDNF expression in the hippocampus. It has been found to reduce amyloid beta accumulation. And in observational human data, higher irisin levels are associated with better cognitive outcomes in older adults. Low grip strength in this framework signals not just reduced muscle mass, but reduced myokin secretion, a reduction in the muscle's active contribution to brain health. These three mechanisms together explain why grip strength is not merely a proxy for overall health. It is a direct input into the brain's maintenance system. Robert's daughter showed him the numbers from his dynamometer. His left hand, 34 kilograms. His right hand, 36 kilograms. For a man of his age and body size, both measurements fell in the adequate to good range above the threshold associated with elevated dementia risk in most normative references. He was not in the lowest quintile. He was, however, lower than he had been at his last assessment 2 years earlier, which his daughter had also measured, because she was the kind of researcher who kept records of Sunday lunch tests. The level matters, she told him, but the trajectory matters more. That sentence, the level matters, but the trajectory matters more, is the most important thing anyone can understand about grip strength as a cognitive predictor. Because grip strength at a single point in time is informative. Grip strength declining over time is a signal that demands attention, and the English longitudinal study of aging specifically found that the rate of grip strength loss predicted the rate of subsequent cognitive decline. Robert's decline was modest. His daughter was not alarmed. But she was interested. She asked him 2 questions. The first, was he still walking every morning? He was. The second, was he doing any resistance exercise? He was not. He had been meaning to. He had not started. This is why, she said, pointing at the dynamometer numbers. This is what meaning to looks like. The evidence on resistance exercise and grip strength in older adults is extensive and consistent. Multiple randomized controlled trials have found that progressive resistance training specifically targeting the muscles of the arm, shoulder, and hand produces significant improvements in grip strength over 8 to 12 weeks in older adults. And the same training has been shown to increase BDNF, reduce inflammatory markers, improve sleep quality, and support gut microbiome diversity, the same downstream benefits as the aerobic exercise we covered in video 11, through complementary mechanisms. Robert started twice weekly resistance training with a trainer 4 weeks after that Sunday lunch. Simple exercises, grip strengtheners, farmer's carries, band resistance work for the forearms. 3 sets, 30 minutes. At his 3-month reassessment another Sunday lunch, another dynamometer, his left hand had returned to 37 kilograms, his right to 39. Both hands stronger than 2 years earlier. He said to his daughter, "I thought I was measuring my hands. Turns out I was measuring my brain." Here is what you can do with this information. The first thing, measure your grip strength. A dynamometer, a hand grip measuring device, costs between $20 and $40 and is available from most pharmacies or online. The test takes 30 seconds. The measurement matters. The second thing, track it over time. Robert's daughter had kept records of every Sunday lunch test, not because she expected a crisis, but because she knew a single measurement tells you where you are, and a series of measurements tells you where you're going. Measure now. Measure again in 6 months. The trajectory is the signal. For men over 60, a dominant hand grip strength below 26 to 32 kilograms is generally considered below the clinical threshold associated with elevated risk. For women, typically below 16 to 20 kilograms. But more important than any single threshold is whether your number is moving in the wrong direction. The third thing, if your grip strength is declining, bring the specific number to your physician or a physical therapist, as Robert did, 4 weeks after that Sunday lunch. Not a report of feeling weak. The measured number and the trend. Ask specifically about progressive resistance training targeting grip and upper extremity strength, and ask whether any of your current medications might be contributing to the decline, because anticholinergic medications reduce muscle coordination through the same muscarinic blockade they apply to the brain, and a pharmacological contributor to grip decline won't respond to resistance training alone. Here is the question to bring to your next appointment. I've been tracking my grip strength, and I've noticed it's declining. I understand that grip strength loss rate is one of the most consistent predictors of subsequent cognitive decline in the research literature. Given my current health profile, would a progressive resistance training program be appropriate? And is there anything in my current medications or health conditions that might be contributing to the decline? That question names the finding, the mechanism, and the intervention. And the medication follow-up connects this video to everything else in this series, because the anticholinergic burden we've discussed throughout, from Margaret's combined score of 9 to Francis's sister's oxybutynin, reduces muscle coordination and brain function through the same pathway. Grip strength decline may be the first visible sign of a burden that hasn't yet shown up on a cognitive test. Robert's daughter stayed for dessert, and for coffee, and for a walk around the block after, which Robert had insisted on because he was doing an outdoor walk every morning and he saw no reason Sunday afternoon should be different. She said, walking beside him, when I was in graduate school, we used grip strength in studies to estimate cognitive risk in cohorts too large to test individually. A proxy measure, we called it. I never thought I'd be doing it at my father's kitchen table. Robert said, "Is it working?" She said, "I don't know yet. That's why I keep coming back." In the next video, I want to cover something that connects to what Robert's daughter just said, the difference between a proxy measure and a direct one. Because there is now a direct test of brain biological age, not cognitive performance. Not risk markers, but an actual measurement of how fast your brain is aging at the cellular level that is moving from research into clinical accessibility. And what the early data shows about who ages fastest and why is one of the most striking findings this channel has yet covered. Subscribe so you don't miss it. And if you don't know your grip strength and you haven't tested it, and you've been meaning to find out, it takes 30 seconds, and it might tell you more than you expect. I'm Dr. Ben. I'll see you in the next one. The information in this video is for educational purposes only and does not constitute medical advice. Always consult your doctor or a qualified healthcare professional before starting any new exercise program, particularly resistance training. Grip strength measurement is not a diagnostic test and should not be used to self-diagnose any medical condition.